Welcome back to the fourth video reviewing the basics about T cells. So far, we have built a simple framework. T cells do not usually recognize whole pathogens directly. They recognize peptides displayed on MHC molecules. We also learned that MHC class 1 usually points towards CD8 T cells, while MHC class 2 usually points towards CD4 T cells. Now we are ready to ask the practical question. What do CD4 and CD8 T cells actually do? The short version is this: CD4 T cells coordinate, CD8 T cells eliminate. Different roles, same mission: protect the host. Hi, I'm Christopher, and in this video we're going to compare CD4 and CD8 T cell responses using a very practical clinical question. Where does the pathogen live? That question helps predict which immune tools matter most. If the pathogen is inside a host, especially in the cytosol, then CD8 cytotoxic T cells become very important. They can recognize infected host cells displaying peptides on MHC class 1 and help eliminate those infected cells. If the pathogen is outside host cells, then CD8 killing is not usually the right tool. Extracellular bacteria, parasites, toxins, and free viral particles are better handled by antibodies, phagocytes, and CD4 helper T cell coordination. And if the pathogen is inside a vesicle, such as an intracellular bacterium surviving inside a macrophage or epithelial cell, then CD4 Th1 responses become especially important because they activate macrophages to improve intracellular killing. So the key idea is not just CD4 versus CD8. The key idea is match the immune response to the pathogen's location. Let's start with an intracellular virus. Viruses are classic intracellular pathogens because they must enter host cells and use the host cell machinery to replicate. Once viral proteins are made inside an infected host cell, those proteins can be processed and displayed on MHC class 1. That display tells CD8 cytotoxic T cells, hey, something's abnormal and happening inside this cell. A CD8 T cell recognizes viral peptide MHC1 complex, binds the infected cell, and delivers a cytotoxic hit. Perforin and granzymes help trigger cell death of the infected cell. That matters because killing of the infected cell helps stop viral replication at the source. But CD8 T cells are not working alone. CD4 T cells help support the response, strengthen CD8 activation, expansion, persistence, and memory formation. They can also support antibody responses that help neutralize free viral particles outside cells. So for an intracellular virus, the key pattern is CD8 does the killing, CD4 helps strengthen and coordinate the response. Now let's look at the same general pathogen category from a different angle. Viruses do not spend their entire life cycle inside cells. At some point, virus particles may be free in extracellular fluid, moving between cells and trying to infect new targets. That is where antibodies matter. Antibodies can bind free virus particles. They can block attachment and entry into host cells. They can neutralize infectivity. They can also help tag virus particles for clearance. But strong antibody responses often depend on CD4 helper T cell support. B cells can bind antigen, but to generate a strong, high-quality class-switched antibody response, they usually need help from CD4 T cells. That help includes cytokines and contact-dependent signals interacting on B cells. So the important distinction is this: virus inside the cell requires CD8-mediated killing of infected cells. Virus outside a cell can be neutralized by antibodies, and CD4 T cells help drive that antibody response. This is why viral immunity is not simply CD8 or antibody, It is often both, depending on where the virus is in its life cycle. This is where it's easy to get tripped up. It's tempting to say, if it's intracellular, CD8 must be the answer, but not always. Some intracellular bacteria survive inside vesicles or phagosomes rather than freely in the cytosol. Chlamydia felis is a good veterinary example because it survives inside intracellular inclusions. When bacterial antigen is in vesicular compartments, it's more naturally processed through the MHC class II pathway. That means CD4 T cells, especially T helper 1 or Th1 cells, become central. A macrophage or infected antigen presenting cell displays bacterial peptide on MHC class II. A CD4 Th1 cell recognizes peptide MHC2 complex and releases cytokines, especially interferon gamma. Those Th1 cytokines activate the macrophage. Activated macrophages become better at killing what they have engulfed. They can increase phagolysomal maturation, reactive oxygen species, nitric oxide production, lysosomal enzymes, and acidification. So for vesicular intracellular bacteria, the key pattern is this. Inside of a vesicle leads to MHC2 and CD4-Th1 and ultimately macrophage activation. That is different from the cytosolic virus pattern. This slide addresses a common misperception. Why can't CD8 T cells control extracellular bacteria? Because CD8 T cells need the right target. CD8 cytotoxic T cells recognize peptides presented on MHC class I. MHC class I displays peptides from inside nucleated host cells. That means that CD8 T cells are designed to identify and kill infected or abnormal host cells. But extracellular bacteria are not inside host cells. They are not generating bacterial peptides in the host cell cytosol. And they are not producing a classic peptide MHC1 display on an infected host cell. So CD8 T cells do not have the right target. And even if a CD8 T cell is fully functional, its job is contact-dependent killing of host cells. A free bacterium in tissue fluid is not an infected host cell to be eliminated by cytotoxic killing. The better tools are CD4 helper responses, antibodies, macrophages, and neutrophils. So the bottom line is this. Outside the cell equate to CD4 help, antibodies, and phagocytes, but not CD8 cytotoxic killing. Now let's anchor this in veterinary examples. First, CAV2, canine adenovirus type 2. This is a viral infection. When a virus is replicating inside epithelial cells, CD8 T cells are important because infected cells display viral peptide on MHC class I. But free virus particles outside cells can still be neutralized by antibodies, which depend on CD4 helper support. So CAV2 is a great example of an integrated response. CD8, CD4, and antibodies matter. Second, Chlamydia felis. This is an intracellular bacterium that survives in intracellular compartments. Because the bacteria is associated with vesicular compartments, CD4 Th1 macrophage activation becomes especially important. The T helper cell is not just helping in a vague way, it's activating the infected or responding phagocyte to kill more effectively. Third, Staphylococcus pseudointermedius. This is an extracellular bacterium in common clinical contexts, such as skin, ears, and wounds. Here, the key immune team is CD4 help, antibodies, macrophages, and neutrophils. Antibodies can opsonize bacteria, phagocytes can engulf and kill them. CD4 T cells coordinate and strengthen the response. To review, CAV2, intracellular virus, CD8 killing, plus CD4 and antibody support. Chlamydia felis, Intracellular bacterium in vesicles means CD4-TH1 macrophage activation. And Staph pseudo intermedius, an extracellular bacterium. That means CD4 help, antibodies, and phagocytes. Different pathogens, different niches, same principle. Match the response to the location. Let's finish with the summary table. If the pathogen is an intracellular virus, like CAV2, the main problem is that the virus replicates inside host cells. The key response is CD8 killing, supported by CD4 help and antibodies for free virus. If the virus is free or extracellular, the main problem is spread to new cells. The key response is antibody neutralization, with CD4 helper T cells supporting B cell antibody production. And if the pathogen is an intracellular bacterium in vesicles, like Chlamydia felis, the main problem is persistence inside vesicles or inclusions. The key response is CD4-TH1 activation of macrophages. And if the pathogen is an extracellular bacterium, like Staphylococcus pseudointermedius, the main problem is that bacteria remain outside host cells. The key response here is antibodies, phagocytes, and CD4 help. So play it again, Sam. Location predicts response. Pathogens inside host cells often need CD8. Pathogens outside cells or in vesicles often need CD4 help, antibodies, and phagocytes. Thank you for watching. I'm Christopher Lee, and what you understand today helps you protect patients tomorrow. And I'll see you in the next video.